I now call to order the Society's 2,425th meeting in the 149th year since its founding on March 13th, 1871. Good evening, everyone, and welcome to the second installment of the fall lecture series produced by PSW Science. These are unusual times, and because of COVID-19, the Society is bringing these meetings to you via Zoom from locations all around the globe, rather than from our usual home, the John Wesley Powell Auditorium of the Cosmos Club in Washington, D.C. COVID-19 has affected us all and is much on our minds. Tonight, we'll hear about work towards bringing this pandemic under control and to a speedy end. Our speaker, Davey Smith, is a leading scientist and clinician who has been working to find ways to deal with the pandemic and hopefully end it. I'm Larry Milstein, President and Program Director of PSW Science, one of the oldest scientific societies of Washington, D.C. PSW Science is a 501c3 nonprofit education and science organization whose mission is to provide a forum to exchange scientific ideas, further scientific understanding, and encourage scientific inquiry. This lecture is being recorded and will be posted to the PSW Science YouTube channel, where it will join over 130 other recordings of PSW Science lectures. We invite you to take a look and explore these presentations. And if you like what the Society is doing, we invite you to become a member and make a contribution through the PSW Science website, www.pswscience.org. The Society is grateful for the sponsorship of the 2020-2021 lecture series by the Public Policy Studies Organization in cooperation with the American Public University, and a donor who has asked to remain anonymous. I am pleased to announce, before we turn to the lecture, it is the tradition of the Society to welcome new members and read the minutes of the previous meeting, summarizing the previous lecture. I am pleased to announce the following new members: Sarah Heap, an astrophysicist retired from NASA, interested in cosmology, space sciences, and instrumentation. Andrea Meredith, professor of physiology at the University of Maryland, with broad scientific interests, who comes to PSW on the recommendation of Anne McQueen, PSW social media director. Catherine Taylor, a kinesiologist interested in space and physics, especially quantum physics, who learned of PSW at an AIAA APU chapter meeting. And tonight's speaker, Davey Smith. We welcome them to membership. Recording Secretary James Seeley will now read the minutes of the 2,424th meeting and the lecture by Charles Clark. His lecture, Vortices of Light and Matter Waves, is available on the PSW Science YouTube channel. James, the screen is yours.
Good evening, everyone. On September 11th, 2020, by Zoom video conference broadcast on the PSW Science YouTube channel, President Larry Milstein called the 2,424th meeting of the Society to order at 8:02 p.m. Eastern Daylight Time. He announced the order of business, summarized the annual report of the Society, and welcomed new members. The recording secretary then read the minutes of the previous meeting. President Milstein then introduced the speaker for the evening, Charles W. Clark, fellow at the National Institute of Standards and Technology and fellow at the Joint Quantum Institute at the University of Maryland. His lecture was titled Vortices of Light and Matter Waves, a new twist on light and particles. Clark began by discussing the nature of light. He summarized Newton's prism experiments, dispersing and recombining rays of light. He noted these experiments are the basis of multiplexing and modern optical fiber telecommunications. He then explained conservation of momentum and superposition of waves. These two principles combine to make interference. Interference enabled experiments that determined the wave nature of light and the independence of light's speed from the motion of an observer. Interference also enables interferometers to accomplish the multiplexing and demultiplexing operations necessary to operate modern fiber optic networks. More recently, interference enabled the Laser Interferometer Gravitational Wave Observatory, or LIGO, to observe gravitational waves. Clark then discussed twisting light. He described the 1990 holograms of Vaznetsov, and Soskin, which used an interferometer and optical fibers to produce laser beams with wave front screw dislocations. Clark then explained plane waves, vessel beams, and orbital angular momentum, or OAM. OAM may produce a corkscrew of light around a central axis, generated by using a spiral phase plate. OAM may also be seen in phased particle waves. Clark said much of the interest in OAM states of light arises from their usefulness in multiplexing data. The same ideas can be applied to ultrasound for communication between underwater sites. And researchers at the University of Waterloo are also using vortex states of light to determine whether they can be used to correct vision or diagnose ocular emissions. Clark then described the mystery of quantum interference as illustrated by the double slit experiment, by which it appears a particle takes two paths. He then explained a series of experiments performed at the NIST Center for Neutron Research. Neutrons have no electric charge and only interact with nuclei through the strong interaction force. They also propagate light within materials. These characteristics allow neutrons to penetrate matter and enable scientists to see through objects. Applying the principles of optical interferometry to phased matter, scientists have developed neutron interferometers. NIST scientists have experimented with applying the ideas of twisted light to neutron beams. To put quantized magnitudes of angular momentum into neutron beams, it was critical to tailor phase plates such that step values were the same. The resulting interferograms showed that angular momentum was transferred to the neutron waves. NIST scientists have also made forays into neutron holography and multiplexing phase plate assemblies by using magnetic fields as a means to create a rectangular pattern of neutron beams with OAM. After the question and answer period, President Milstein thanked the speaker, made the usual housekeeping announcements, and invited guests to join the society. At 9:31 p.m., President Milstein adjourned the meeting. Temperature in Washington, D.C., 23 degrees Celsius. Weather, mostly cloudy. Concurrent viewers on the Zoom and YouTube live stream, 90. And views on the PSW Science YouTube and Vimeo channels, 341. Respectfully submitted, James Heelan, Recording Secretary.
Thank you, James. The minutes will be posted to the website in due course. Please send any corrections or comments on the minutes to corresponding secretary Robin Taylor at pswscience.org. We now turn to tonight's lecture on COVID-19 treatments and trials. And it's my pleasure to introduce tonight's speaker, Davey Smith. Davey is Vice Chair of Faculty in the Department of Medicine, Chief of the Division of Infectious Diseases and Global Public Health, and Professor of Medicine at UC San Diego. He also serves as co-director of the San Diego Center for AIDS Research. Davey has pursued virology research throughout his career. He has been deeply involved in the study HIV transmission and reservoir dynamics. Shortly after learning about the new coronavirus, he refocused his entire research effort on COVID-19. In March of this year, he instigated partnerships across all NIH centers for AIDS research to pool equipment, methods, and knowledge to study COVID-19. Currently, he is involved in numerous COVID-19 clinical studies. Davey was named HIV Researcher of the Year and has been elected Fellow of the American Society of Clinical Investigation and Fellow of the American College of Physicians. Davey earned his MD at the East Tennessee State University School of Medicine. All questions will be fielded after the lecture during the Q&A session. Davey, the screen is yours.
Thank you, Larry. Thank you very much for inviting me. I'm going to be talking about the trials and tribulations of coronavirus, CoV-2, or Junior, as I like to say, of COVID-19. And the question today I want to pose is, can we treat our way out of a pandemic? And a little bit of a hint, we are doing it now. So just to give a quick update is that we should have seen it coming. We've had tons of recent epidemics and viral epidemics in my lifetime. 1981 with HIV/AIDS, which is what I mostly work on, but SARS-CoV-1, or Senior as I like to call it, happened in 2003, then swine flu, avian flu, MERS, Ebola, the co-epidemics of HIV and hepatitis, Zika, etc. And then in 2019, we had SARS-CoV-2. But where are we right now is the good question. Here's the state of the epidemic from a couple of days ago. And you can see it's all across the world. We have almost 32 million cases. We have almost over 900,000 global deaths. We're at over 200,000 deaths in the United States. So it is a deadly pandemic for sure. But I'm going to talk a little bit about epidemic control. And at the beginning of what we can think about, how do we actually control this pandemic? So I'm going to start off with a question. There won't be a test at the end, but it's good for us to think about. What proportion of susceptible people in a population need to be protected? And you can think of masks or vaccination or maybe even treatments. If the R naught, which is the reproductive rate of CoV-2 in the community, is 4. Is it 1 out of 4? Is it half? Is it 3/4? Or does everybody have to be protected? Hopefully at the end of this lecture, you'll have the answer to that question. And a little bit about R naught, which is the reproductive rate. And when we talk about public health and epidemiology and epidemic control, it's really a good, useful equation where C is the number of contacts the infectious person makes per day, per week, per month. P is the probability of transmission per contact, and D is the duration that the infected person is infectious to others. And here's a bunch of infectious pathogens, and their reproductive rate is on the y-axis. Or for like Ebola, for every one person who's infected with Ebola, they could give it to 2 other people. CoV-2, it's somewhere between 3 to 5. Diphtheria is a little bit more, and then you can see that pertussis and measles are up around 15 to 20 people. So for every one person who has measles, if there is enough susceptible people in the population, like everybody's susceptible, then it can get up to 20 people measles. Very infectious. So what are our current efforts about dropping that reproductive rate down to less than 1? So getting to 0 new infections, we have a pandemic toolbox based on this equation. And right now, we're all fixated on quarantines, physical distancing, and bubbles. Our little bubbles of like my household only works with this other household, and that's our social bubble. And that's really based on the concept of decreasing the number of contacts that people have. And that works to slowly decrease R naught. Other ones are like testing. So if I test and I find that I'm infected and I go self-isolate, that decreases the number of contacts that I can make afterwards. So that's also in C. Also, if I test and I'm found to be positive and somebody goes out and contact traces other people to find other people who are positive and they self-isolate, that also decreases the contacts. Other things that people talk about a lot is masks. And you can think of masks. If I wear a mask when I am infected, then that decreases my infectiousness to somebody else. So that's the probability of transmission. If somebody else wears a mask and they don't have the infection, but I do, that decreases the chances of them getting the infection. So it decreases both infectiousness and susceptibility. Another one that is aspirational at the moment is antiviral treatments. And you could think of if I were to be infected and I take an antiviral medication and it decreases the amount of virus that I have in my nose, and that means I'm less likely to give it to somebody else. So that decreases the probability of transmission. It also decreases the duration of my infectiousness, which is the D in the equation. Other ones, perhaps we can use those same treatments for prophylaxis so that you can give it to somebody to keep them from getting infected if they were to be exposed. And there are trials going on right now in nursing homes giving people potential antiviral treatments to protect them. Both of these have been used in the HIV epidemic for a long time, treating people who are infected to decrease their virus so they don't transmit it, but also give medications to people who might be exposed to prevent them from getting it, also called PrEP, or pre-exposure prophylaxis. And of course, one of the things that lots of oxygen in the air at the moment, people talking about a lot, are vaccines. And vaccines are really meant to help people become immune, so it decreases their susceptibility of an infection. And at the moment, the FDA said they would be happy if they had a vaccine that reduced that risk by 50%. So that decreases susceptibility or the probability of transmission by 50%. Just to note, though, if I had a vaccine that worked 50% of the time and I go into the hospital, I'm an infectious disease doc, and I see somebody and I get exposed for whatever reason to a coronavirus and I had been vaccinated, there's still a 50% chance that I might be infected, in which case I would really like there to be an antiviral treatment in case I needed one. So let's just answer my question at the beginning. So if I have an infection that has an R naught of 4, so for every one person who's infected, if there's a totally susceptible population, they would give it to 4 other people. If I were able to stop that transmission, decrease the susceptibility in the population by 3 out of 4, then only one of those infections would get across. And then in that same population, the 3 out of 4 people are protected, then eventually that infection would die out. So the answer at the beginning was, if I have an R naught of 4, then I need to get about 75% of the population protected in some way to have that epidemic, that pandemic go away. So now I'm going to talk a lot about antiviral treatment and how we might be able to use that as a way to get us out of this pandemic. I'm going to start off with a natural history of the coronavirus infection. So somebody gets exposed to the virus, and everybody who gets infected starts off with no symptoms. But most people go on to develop mild symptoms, and some people go on to develop moderate symptoms, and even fewer people go on to develop severe symptoms. But that's the natural history. At the beginning, it's all about the virus. So the virus comes in, the virus levels go up, and then they go down, actually, while the natural history of the disease actually becomes more severe. So if I'm going to try to stop this course of this natural history of infection at the very beginning, then I really need to focus on the virus, and that's to use antivirals. So when we first started this, we said, okay, is there an old drug that we could repurpose for this effect? And here is a diagram of the virus. So the virus comes in and it attaches to this first little receptor called ACE. And we have these medications called ACE inhibitors, and they're currently in the trial to see if they can block that virus from interacting with that protein that's on those cells to keep it from attaching. So those trials are going on at the moment. The next one is then the virus comes in, it forms this little cup in the cell membrane, and it goes into this vacuole, and it needs a host protease to get out of that vacuole. And there are protease inhibitors that can do that. And one of those is called Camostat, and that's currently in clinical trials at the moment as well. But let's say if the virus is able to stay in that vacuole and perhaps get out. One other medication that we talked a lot about early on is called hydroxychloroquine. And we know that hydroxychloroquine can acidify that vacuole and thus not allow that virus to get out. And that was the proposed mechanism there. And I'm going to talk about hydroxychloroquine in a second. But let's say it does get out, it makes a polyprotein. That protein needs to be broken up, and it does that with other proteases. And we have protease inhibitors from the HIV world that have been tested to try to break that part of the virus life cycle up. The other part is the virus needs to make copies of its genetic material, and it does that through nucleases and synthetases and polymerases. And that process, the polymerases, the viral RNA polymerases can be blocked as well through enzyme inhibitors. And many of those have been tested, including you might have heard of one called remdesivir. But other ones like ATIA, AT527, which is a hepatitis C drug, may also have activity, and it's currently also being trialed. So let's talk about hydroxychloroquine just a little bit. So back in December of last year, was on a nice vacation with my parents and heard about the coronavirus breaking out in China. And I was like, oh, this is going to be a problem. But at the same time, well, about a month later, we had some data showing that hydroxychloroquine and chloroquine here on this first graph actually inhibited the viral growth. And they compared it to remdesivir, which inhibited the viral growth as well, but not as well as hydroxychloroquine. So the chloroquine or hydroxychloroquine inhibited it both throughout, at entry and post-entry, while remdesivir really only did it at the post-entry site. So that was all well and good in cell culture. But now they have, other people said, okay, well, let's look at it in people. And this is the French study. And on the y-axis is the amount of virus that somebody has in their nose, or the number of positive samples. And at the beginning, the people who got the placebo, they all pretty much had viral detected in their nose. But people who got hydroxychloroquine here in the blue line went down. They had more samples that were negative of the virus. And people who got hydroxychloroquine plus azithromycin really went down much faster. And what's interesting about that is hydroxychloroquine acidifies that vacuole, but in fact, azithromycin also acidifies that vacuole, maybe keeping that virus in. So myself and others said, oh, well, we really ought to try this. And we had actually been in touch with the NIH before. And here's just a little timeline, is that in November, Junior breaks out, hydroxychloroquine works as a concept. I submit it to the NIH. We have a discussion, but they don't really move on it too much. Back in March and April, the French study comes out and our President Trump starts touting hydroxychloroquine. And this is really the time that we should have intervened to really do the science to say whether or not hydroxychloroquine works or not. Because afterwards, the FDA gave an emergency use authorization. And what that meant is that any provider, any physician could just give hydroxychloroquine without really knowing that it works. And in fact, that's what happened. There was massive use of hydroxychloroquine for people who were already sick. And if you remember that natural history, hydroxychloroquine actually works on the antiviral side and not in the severe stages. So in fact, they were giving the medication in a setting which probably really didn't have much to do with the virus, but had to do more with the inflammation. But at the same time, I got a call from the NIH and said, hey, we really understand that we need to do that trial. And then, so we got it together, we got a big trial up and running, and about that same time, papers started to report that hydroxychloroquine is dangerous, definitely in the setting of an inpatient trial where in the inpatient setting where people were already sick, and then the papers got retracted. And so the study opened and it closed with only 20 people enrolled. And the reason it wouldn't enroll, it was just too much circus out in the media and social media about hydroxychloroquine. But then the FDA went ahead and revoked the EUA because there was no data to show that hydroxychloroquine worked or that it didn't work. What's interesting is this study, which is from the Henry Ford Foundation, and this is a Kaplan-Meier curve. So on the y-axis is the survival probability, and the green line is people who got hydroxychloroquine early, and then the red line is people who didn't get hydroxychloroquine, but the people who got it earlier were more likely to survive. The issue was these are small numbers and really weren't able to figure out whether hydroxychloroquine should be used in all instances for outpatient disease. And why that's important is it was probably our best chance to use a drug that we knew was relatively safe in a very safe setting, in an outpatient setting, that was going to be cheap. Everything else after that was probably going to be expensive. So the lessons learned that I want to talk about today is don't fall into the trap of something must be done, this is something, so this must be done. Do the rigorous science and don't politicize the science. Another drug that was repurposed that you might have heard about is called remdesivir, and it works at this late stages of where the virus has to replicate its nucleic acid, so at the polymerase, and it blocks that enzyme to keep it from happening. What's interesting here is that this drug was made for SARS-CoV-1 senior and another coronavirus called Middle Eastern Respiratory Syndrome, or MERS. So that was already on the shelf. It was owned by Gilead. So there wasn't a way for a whole bunch of doctors to use it. It was only in the hands of Gilead. And Gilead, the company, said, you know what? We're going to do this clinical trial to see if it actually works before we just let a lot of people use it. And this is the trial that they did. And on the y-axis is the proportion of people recovered, and the x-axis is the days. And you can see that remdesivir did diverge from placebo a little bit. I wouldn't say that that was strong, but for hospitalized patients, remdesivir helped them recover better than placebo. So that was a good lesson. We now have a drug for people who are in the hospital. Remdesivir has to be administered in the IV intravenously multiple times a day because its half-life is short. So it's not useful outside of the hospital, but at least in the hospital it had some effect. Another drug that was on the shelf is called EIDD 2801. It's now bought by Merck, and it has a different name, but it was made by a company, Ridgeback, for Ebola. And it specifically works on the RNA machinery to make duplications of itself. And it causes the virus to make lots of fatal errors when it tries to duplicate it. And it duplicate RNA. So it works on lots of RNA viruses like the flu, Ebola, other coronaviruses, etc. And here, when they tested it in MERS in vitro, this is the percent inhibition. It has a nice curve at relatively low levels. It's not quite as good for CoV-2, but still does inhibit CoV-2 at least some. And this is now being in clinical trials for Merck. So what about a new drug? So those are all repurposed drugs. Can we get a new one? Well, we can. Let's just go back in time and think about the first Nobel Prize ever, which was for diphtheria. And what scientists did at that time is they took diphtheria, they injected a horse with diphtheria, the toxin from the diphtheria. The horses mounted an immune reaction, which was an antibody. They collected the sera from the horses and they concentrated it and came up with an antitoxin medication so that when a kid here at this time, got diphtheria, they could get the antitoxin and then survive. So the question is, could we do something similar for CoV-2? So antibodies as therapy is what I want to talk about. And here you can have one mechanism where the antibody that recognizes the virus blocks it from getting attached to its receptor ACE2, or it can perhaps get into a cell and block that virus from getting out of that vacuole. The other one is that it can get on this FcR receptor and attach to the virus and keep it localized within the vacuole so that it goes down to a destruction pathway versus a get out of the virus pathway. And you might have heard that people are using these therapies in terms of convalescent sera. So someone got infected with the virus, they mounted an immune response, they did okay, but they made some antibodies, they give us some blood, we take out the antibodies in the form of plasma, we return their red blood cells, and then we use that plasma for someone who might be very sick with the virus and hope that it can fix them. And there's some data that there might be some signal there. So here is a JAMA paper that was earlier this year that showed here on the y-axis is the improvement rate for all patients, and that convalescent plasma had some efficacy rather than control. And even in severe disease, it looked like that there was a signal of improvement. In life-threatening disease, there was no improvement. This study didn't reach its endpoint. It was underpowered and stopped early. So we don't really know from this study whether or not it worked. And this brings me to my next point, because the FDA, based on some of these small studies and little data, granted convalescent plasma, again, an emergency use authorization, despite there being rigorous clinical trial data. And I'm just worried that this falls back into the trap of something must be done. This is something, so this must be done. I really encourage us all to think about doing rigorous science, get the real answer, find the real drug, and don't politicize as we move forward. So there's another thing that we can do, and this is called monoclonal antibodies. And this is something that we worked in my lab, but others have done it as well. And basically, there were people who were infected with coronavirus, and they developed really strong antibodies. In fact, there was some antibodies that were super potent. And we could pluck those antibodies out. We can put those into cells called hybridomas. We can grow up those cells, purify them, expand them, make them give us lots of this one antibody, so the monoclone, one clone, and turn it into a treatment. And it's been done for Ebola and HIV, respiratory syncytial virus, etc. So we had the proof of principle that it could work, so that when you give it to somebody who's infected, hopefully it treats them, but it also might protect somebody who gets exposed to the virus. And there are 25 companies, maybe even more, who are currently going in this route. Other ones are cows. So here is a company called SAB Pharmaceuticals, and they take cows and transplant in their immune system with a human immune system. And they expose those cows, like we did with the horses before, to the coronavirus antigens, and that causes the cow's human immune system to mount an antibody response, but it's polyclonal. So it's not just one antibody, but it's lots of different antibodies. And then the cows make lots of plasma, and that could be then used for treatment. And this is currently being studied. So back to my question of when to treat and what to use. So here at the beginning, we'd want to use an antiviral. But later in the stage, what's going on there? Well, it's all about the inflammation. So the virus causes the immune system to overreact, and that causes severe inflammation in some people. And that inflammation can be so severe that it can be life-threatening. So people have started to look at immune modulators to fix that. And this slide is totally meant to overwhelm you because there are so many different pathways for inflammation that this virus might take. And many of these have various drugs that have already been created to stop that inflammation. And many of these are already being tested for coronavirus, including glucocorticoids, mineral corticoids, anti-cytokines, JAK-STAT inhibitors, etc. And all these are currently being tested for the severe late stages of disease for coronavirus. And here's another big trial called the recovery trial from the UK, and they tested a few things. And one of those things they tested was dexamethasone that seemed to work. Other ones that didn't work were like lopinavir, ritonavir, and hydroxychloroquine, which is in sick people in the hospital. Azithromycin is still pending. Tocilizumab didn't work here, and convalescent plasma, despite being tried for a long time, still hasn't shown efficacy. But here is dexamethasone, and percent mortality is on the y-axis, and people who got regular care, what they call usual care, died at a pretty high rate. So this is people dying actually in the intensive care, and dexamethasone decreased that risk of death in that group. Steroids recently were reported that might also have an effect, which is similar to dexamethasone, and this is the cumulative proportion of people who didn't need organ support, so they didn't need a ventilator. And people who got usual care needed the ventilator more, that's in the black line, versus those people who got some form of hydrocortisone or steroids needed less of organ support, like ventilation. It didn't quite trigger superiority, but you can see that those lines are decreasing. So the next thing they did is they said, okay, well, we don't have definitive proof, but perhaps we can do a meta-analysis. And they took all these different trials over here with dexamethasone, like the recovery, the Dexa-COVID, hydrocortisone, methylprednisone, and they said, okay, what is favored or not favored? And they put all these studies together, and pretty much all of them are on the left side, which favored the use of steroids, versus on the right side, which did not. Okay, we're back to when to treat and what to use. So we have the inflammation and some immune modulators. But another weird thing about this virus is that it causes what we call a hypercoagulable state. And this is just where the virus causes lots of clots to form in the body. They form in the heart, they form in the brain, they form in the lungs, and that causes a lot of morbidity and mortality. And people are now looking at anticoagulants as therapy for SARS-CoV-2 infection. And this could be low molecular weight heparin or regular heparin or direct oral anticoagulants. And it's basically trying to hit all these different components of the clotting cascade to prevent this clot formation. And we've known for a long time that inflammation and coagulation go together, but this virus seems to be particularly good at that, or bad at that, depending on how you look at it. The next thing I'm going to talk about is a thing called Operation Warp Speed, or Active-2. And the reason I'm going to talk about it is I'm the chair of Active-2. And the question is, how do we get to therapies as quickly as possible? And ACTIVE stands for Accelerating COVID-19 Therapeutic Interventions and Vaccines, ACTIVE. And it's based on April 17th NIH public-private partnership to make these therapies and vaccines as quickly as possible. And here are all the treatment trials. So Active-1 is immune modulatory, Active-2 is outpatients, Active-3 is inpatients, Active-4 is anticoagulants, and Active-5 are the inpatients and combinations of medications. The only two that are currently running at the moment, the other ones haven't got off the ground yet, are Active-2 and Active-3. So the Active-2 objective is to rapidly and efficiently evaluate multiple potential therapeutics for COVID-19 in an outpatient setting. So we want to catch people as early as possible and keep them from getting into the hospital. Our trial is designed as a randomized blinded controlled platform that allows multiple agents to be added and dropped during the study. It begins with phase 2, followed by a larger phase 3 for promising agents. When 2 or more new agents are being tested at the same time, we can use the same placebo if feasible. This increases the efficiency of the trial. And basically it works like this. So you have phase 2 that starts off, and it starts off with agent A. We enroll 110 people and give them agent A. We also give 110 people a placebo. If it shows no promise, we stop with that drug, and perhaps agent B comes in, the same thing with 110 people. If it shows promise, we go to a much larger trial with 890 people. And at the same time, or a little bit afterwards, we can start with another agent, agent C. And the whole purpose is to find better and then better and then better therapies for COVID-19. The agents are chosen based on their activity against SARS-CoV-2 entry and replication, their pharmacokinetics, their safety data, and their potential to expand to phase 3 if found effective. Our study eligibility are adults with active COVID-2 infection within a week, and they have to have symptoms within the start of the symptoms within the past 10 days, and prior to entry, at least one of the symptoms within the past 2 days. And these symptoms are regular COVID-19 symptoms that we know about, a fever or feeling feverish, cough, shortness of breath, sore throat, etc. We also have a stratification of higher risk of COVID-19 progression. So older people with comorbidities like high blood pressure, cardiovascular disease, or diabetes, or people who are heavier with a BMI greater than 35. We're also looking at whether or not it helps to start early versus late, so before and after 5 days. And then our phase 2 objectives is determine the safety and efficacy of an agent to reduce the symptoms of COVID-19 and to see if it also decreases the amount of virus in somebody's nose. If it does, then we can graduate that compound. So if it can reduce the proportion of negative, if it can increase the proportion of negative nasal swabs by 20%, then we think that that agent is useful and it can move on. Or if it decreases the amount of virus in terms of the log copies, it can move on. Or if it reduces the symptoms by 20%, or if it increases somebody's oxygenation and is still found safe, we say that drug can move from phase 2 to phase 3. We have a lot of interventions at the time. So in phase 2, somebody gets the drug, We take blood, saliva, we teach them how to do a study diary, we do a nasopharyngeal swab, we collect their oxygen saturation, we set them up to do daily reminders throughout 28 days, and then we have them come back in on days 3, 7, 14, 21, and 28 to do all these assessments again, and then we have longer term follow-up at weeks 12 and 24. If it graduates based on symptoms and oxygenation or virology, we do a much lighter touch study for phase 3, where they get the drug, we see them at the beginning, we do the blood collection, the nasopharyngeal swabs, the oxygen, teach them how to do the study diary, teach them how to do their nasal swabs at home, and then we don't see them again until day 28, but they're sending in their nasal swabs during the time and telling us about their symptoms. And then we have that longer term follow-up. The primary objectives for the phase 3 study is hospitalization and death. So we really want to know whether or not people who get the agent are prevented from getting into the hospital or dying compared to people who got the placebo. And if it does, then the FDA has said, we will approve your drug. We are chasing the dragon in the United States for the coronavirus. This is currently what the map looks like. So the darker colors are where the virus is. And we have our study set up that's called Rise Above COVID. And we are putting clinical trial sites where there are the more red, the darker places across the United States. But this is a global pandemic and it's all over the world. And we now have sites opening in Brazil, Chile, Peru, South Africa, Philippines, etc. I do think there's hope, both for the antibodies, perhaps convalescent plasma. Hopefully we can get an answer. The polyclonal animal stuff is interesting. Monoclonal antibodies, I think, will probably be our first ones that are approved. Lilly just came out with some nice data last week showing that there's definitely antiviral activity with their drug, but not enough to say that they can get approval yet. So that's one of the agents that we're testing in our trial. Other small molecules like Camostat and the ones that I talked about earlier, those antithrombotics might be very interesting, as well as immune modulatory drugs like dexamethasone, glucocorticoids, and other medications. So let's just start back where we came from the beginning. We can treat our way out of the pandemic, and we did it in HIV for the most part. We got good therapies, and we started this program called Ending the HIV Epidemic, and it made on 4 pillars: to diagnose, treat, prevent, and respond. And these are the same pillars that we can use for this current epidemic if we actually had a really good treatment that could decrease the infectiousness of people, help them to live better, and not get so sick. So my parting thoughts: good, science works, and we really need a treatment that'll help keep people junior outside the hospital, and it could also help with pandemic control. The bad is that politics is bad for the science. We really are unlikely to have an inexpensive, widely available treatment anytime soon, but hopefully we can come up with them as we move along. These are my collaborators, my Active 2 team. These are the people who gave me some slides that I like to acknowledge. And then I have my disclosures, where I'm a consultant or a scientific advisory board, and then I'm happy to take some questions.
Thank you, Davey. Now we have time for a question and answer period. Zoom participants, raise your Zoom hand, and we will unmute you in turn for you to ask a question. If you are a Zoom participant, but you do not have a microphone, you can also submit questions in the Zoom Q&A panel. And YouTube viewers can text questions in the YouTube chat box. All text questions, whether on the Zoom Q&A panel or the YouTube chat box, will be read aloud for everyone to hear. So let's start with Carl Merrill. Carl? Can you unmic?
Yeah, no, I can hear you now. Thank you.
Great.
No, so one of the questions I had was a relatively inexpensive anti-inflammatory agent that you actually mentioned, and that's colchicine, but then you didn't mention whether there are any studies with it. So I was just curious as to.
Yeah, that's a good question. So there are studies with colchicine going on right now. One of the sites is at UC San Francisco. They haven't read out that study yet. I think it might be, it's probably most useful going to be in the later stages of disease process, but that study hasn't finished yet.
Thank you.
Richard Garwin, if you can ask your question. Richard?
And there was a small study in Spain published end of August in which 50 patients in the treatment arm of the study with calcifediol, a vitamin D3 metabolite, were treated. Only one needed to go to the ICU. Of the non-treatment arm, half as many, 26 patients, 13 had to go to the ICU and 2 died. So my question, if you had early symptoms of the disease, would you take the calcifediol treatment?
Me personally, I would enter into a clinical trial, and it might be one with vitamin D or other supplementation along those lines. I really want to encourage all of us to think about how science can get us out of this pandemic. I totally think that vitamin D and the vitamin D pathways are very important and seem to have some role in the pathogenesis of this virus. It does seem to be more skewed to people who might be more vitamin D deficient at the beginning, so that might help. But we won't ever really know until we do rigorous studies. And the Spanish study was great. And now we need to expand it to much larger studies to really know. And the reason I say that is, honestly, if we had started real trials when it broke out in China, and then it came to Italy, it went to Iran, Spain, etc., came to the US, if we were ready for clinical trials, many of these questions would have already been answered. But at the moment, we're still left unknowing.
There's an NIH protocol for which this was the pilot, dealing with 1,000, but so far as I can see, it hasn't been populated. So it takes a long time. And when we had cowpox for smallpox, didn't understand it, but it worked.
Sure, there are going to be agents that perhaps we don't know at the time how it works. Thank goodness we know how cowpox works at the moment. But just to, one of the things that hurts our ability to enroll clinical trials is people just using something outside the setting of a clinical trial.
Just let me remind everybody who wants to ask a question who's on the Zoom call to raise your hand so we know to call on you. My next question comes from Michael Helton. Mike, are you able to turn your mic on and raise and ask your question?
Yes, can you hear me?
Yes, all good.
Yes, my question is, if a person is tested positive for the virus and then after 2 weeks is tested negative, can that individual still carry and spread the virus?
It's very unlikely for that person to still be able to spread the virus.
I think our positive and negative tests here are viral RNA tests, just to be clear.
The viral RNA tests.
Yeah.
And in reality, that person is probably unlikely to spread it even if that test is positive after 2 weeks. And the reason we think that, we know that they have less infectious virus and that they're making antibodies that probably glom onto the virus. So even though we're able to detect it, it's not as infectious and probably not infectious at all after about 2 weeks in most people. So if they were immunosuppressed and they don't make good antibody, that might be a different story. But most people will make enough antibody after 2 weeks to really keep that virus from being infectious.
Okay, thank you very much.
We have a question from the YouTube chat box from Joel, who's a PSW member. He wants to know, is it known why SARS-CoV-2 causes thrombosis? And is the mechanism understood?
The short answer to that question is no, not fully understood. We definitely know that many hyperinflammatory syndromes like sepsis even causes clotting problems, both people unable to clot and people to clot too much. This particular one looks to hit a pathway called bradykinin. And that pathway increases clotting that happens mostly in small vessels, but it can have big clots as well. So there is more to come out along those lines. And I think the clinical trials that look at those anticoagulants to really tease out that cascade will be very informative there.
Next question will be from Bruce Murray. Can you, yes, can you unmute and ask your question, Bruce?
You mentioned monoclonal antibodies, with which I am not familiar at all, but you said they had been used in HIV, and I don't recall hearing about that, and I'm gay, and I've heard about HIV for a long time. Could you talk a little bit more about that, please?
Yeah, for sure. So people have been making these very potent monoclonal antibodies for multiple viral diseases, including HIV. And they're using it in a way, not so much in terms of treatment for HIV, but to try to make cures for HIV. They think that these monoclonal antibodies can get into deep tissues and eradicate infected cells in a different way than perhaps our regular HIV therapy can. But good question.
Thank you.
I have a question also from YouTube chat box from Don McCrae. Can you provide clarification on if hydroxychloroquine was useful early in the disease or not?
I would love to have the answer to that question because I just don't know. And the reason I don't know is that the studies that were done were not sufficiently powered. The ones that were completed were not sufficiently powered to answer that question. We never did a large enough clinical trial in that population to say that it worked or not. And I'm afraid that we just never will, given the hype and politics of the situation.
That include not only the US, but all over the world?
Yeah, all over the world. It became just a hot mess. I think that there's better data in Barcelona that might be coming out as they finish up some of their studies, and that there is a worldwide consortium led by the Gates Foundation to take as much data across all these trials and see if they can meta-analyze them to come up with an answer. But even then, it's hard because it's going to be putting a lot of disparate trial data together.
And the French never followed up on their initial.
The French did exactly what we did. They let it have an emergency use authorization, and then they reneged it, they took it away. It was just disappointing.
Another question from the YouTube chat from Jerry Krasner. What is the likelihood and treatment implications of the viral mutations? In other words, how rapidly is this thing mutating, and what are the implications, not just for treatment, but I guess even more so for vaccine?
Yeah, that is a great question, and probably could spend way too long talking about it. I do think there are a few things to know. Is that this virus, even though it's an RNA virus, it does not mutate as much as HIV, and it does not mutate as much as hepatitis C. It does mutate, but since it doesn't mutate as much, we think that we have a better chance of getting a vaccine. As you notice, we don't have a vaccine for HIV, we don't have a vaccine for hepatitis C, and the reason is there's so much genetic diversity and mutations that occur with those two viruses. For this virus, it's relatively constrained, and we think that we would be able to make a vaccine for it. So that's the good news. The bad news is it does mutate, and perhaps can mutate even more under selection pressure by a monoclonal antibody or one of those polymerase inhibitors, et cetera, to actually become resistant to those. And all of these trials really need to look for that and watch for it.
Could you elaborate a little bit? Because there's also MERS and SARS-1, and I don't think we have vaccines for those either.
We do not have vaccines for SARS-CoV-1 senior or MERS. I do want to caution a little bit because the vaccine effort for those two was nothing compared to what the vaccine effort is for this current virus. So I think that gives me a little bit of hope. But I remain cautious. I wouldn't say pessimistic, but I remain cautious that these vaccines might show some efficacy early on, but they might not be durable. And the reason I say that is that in regular coronaviruses, we get infected, we get some immune response, and then that immune response goes away enough so that we get infected again the next winter or the next seasonal respiratory virus season.
So we have another question from Dick Garwin. So Dick, you're on.
Sorry. Yes. Well, one of your early slides showed the ACE1 receptor as the docking target, but it's usually stated as ACE2. So was the ACE1 notation just an error? The upper
Yeah, it was wrong for me. Sorry about that.
Okay, thank you.
Thank you for pointing that out.
I'll fix my slide.
Okay, now we have a question from Al Ehrlich. Al, can you ask your question?
Now can you hear me?
Yeah.
The question is, has anyone started to do statistics, so to speak, to sort out the relative importance of all these various cofactors which people talk about so much? You know, hypertension, diabetes, age, they seem very varied. And has there been any progress in trying to understand how these cofactors scientifically play into the disease?
That is a great question. The first part is, are people looking at it? Absolutely, people are looking at it. Do I know of much progress? Not a little bit. I mean, we've had two pretty high profile papers from our group showing that pre-existing exposure to circulating regular coronaviruses do perhaps lead a little bit of immunity down a pathway. But to really understand the hyper-inflammatory responses that we see with people who have comorbidities is still up in the air. The best signals that I can see is that really obesity and diabetes, which are both actually inflammatory conditions of themselves, do tip that balance more so than, let's say, cardiovascular disease or high blood pressure.
And is the main killer of the disease this exaggerated immune response, or there are other mechanisms?
It is exaggerated immune response in the hypercoagulable state. So when people start making blood clots that go to their heart and to their brain, that's a pretty bad thing.
And so that's two different things.
They are definitely linked. Don't get me wrong. They are linked, but some people die just from an inflammatory response and some people die from a blood clot.
Thank you very much.
I guess there's a question coming in by a private channel as to what do you think? I hate to ask you to prognosticate, but do you think there'll be a second wave, and how bad will it be?
So I'll tell you what I'm afraid of. I will tell you that the fall is going to be very tough. The fall is going to be tough as an infectious disease doctor. And the reason it's going to be tough is because we have every winter a bunch of respiratory viruses. So it's not just coronavirus, but it's the flu, it's adenovirus, it's RSV, and they're all circulating at that time. And people are more likely to be indoors and together, which also is right for spreading these viruses. And we won't be able, we being the infectious disease docs who are on the ground, are going to have a hard time distinguishing when somebody comes in. Is this the flu? Is this RSV? Is this coronavirus? And the current tests we have don't all go together. So there's going to have to be multiple tests that happen to distinguish what's actually going on. And that's going to make it harder for us to do contact tracing and isolation, et cetera. And my point there is everybody needs to get their flu shot. and we need to get our flu shot every year, but this year will be very important because it just takes one of the more dangerous viruses off the table, hopefully for a lot of people, and make our fall better, fall and winter better.
I'm going to sneak a little add on to that before getting to Bruce Murray, who has another question, and that is, I thought there were some reports that influenza infections were decreasing among people who had suffered from a COVID-2 infection. Is that held up, or is that just some apocryphal noise on the internet? Or
So the question is whether or not people who had flu more likely to be protected from COVID-2?
or COVID-2 is more likely to protect them from the flu?
Oh, no, I see what you're saying. So early on, there were these reports that people were not dually infected. So they didn't have two respiratory viruses at the same time.
It's kind of the Southern Hemisphere, by the way.
Yeah, in the Southern Hemisphere for the most part, because they were in the winter while we were in our summer. I read those papers. I don't quite believe it because the testing is was so scant in those areas to really be able to discern co-infections. So I think we'll know more about that this coming fall in the United States.
I have a question from Bruce Murray. Bruce, you can ask your question.
Do we have a good idea by now of the ratio of people in the population at large who are infected but non-symptomatic to those who have actually been diagnosed as infected?
Yeah, that's a good question. So how many asymptomatic infections are in the population? And I'm going to get on my little soapbox here and to say, first of all, no, I don't have good estimates. And the reason I don't have good estimates is that we haven't done that great of epidemiology from the beginning. We did not get good age-appropriate cohorts and follow them over time in these hotspots. And what we're left with is a bunch of very biased cohorts. So those ranges of asymptomatic infections go down from 5% to 40%, up to 40% of people having asymptomatic infections. And I don't know where the truth is. It's somewhere in between those two, but those biased cohorts really muddy the water. We need to do better science in terms of epidemiology to figure out that question. And we need to figure it out based on ages, because it is clear that the younger people are less likely to have symptoms. So, but they're more likely possibly to spread it, which is another reason that fall might be kind of rough with colleges and schools in play. But to answer your question, I don't know what the percent is.
Thank you very much. I was afraid of that.
Yeah, it scares me too.
One thing about this virus is there's an awful lot we don't know.
Yeah. I mean, we've only had it for, what is it? September, so 10 months.
There's been no attempt to just look at the population at random and take a big sample and just test. What is that called? Surveillance testing?
They do these random spot testing. So they take the group and they do all at once sampling. And those are probably our best. The only problem with that is that even in hot spots, the relative proportion of people who are infected are small. So let's say San Diego, we have a 5% rate. You sample 1,000 people, you're getting down to 50 to, less than 100 people still. It just, those small numbers can skew a lot on symptoms and stuff.
We have a question from Gail Milstein. Gail, can you ask your question?
I was wondering about whether or not famotidine and melatonin have been shown to have a favorable effect on treatment. I know there's been a few studies on that.
Yeah, good question. So famotidine, H2 blocker, has shown some efficacy in smaller studies, and those have been published, or at least reported. But bigger studies of famotidine haven't shown much efficacy in those, which just becomes a problem in the literature for negative selection bias for publication. If you show a negative result, you're less likely to put it out in the literature. So I would still take famotidine with a grain of salt at the moment, but those studies are ongoing, mostly in New York, actually. Melatonin, I don't know a lot of, there hasn't been a lot more data published since after the New York outbreak about melatonin. I do know people who are still looking at it, and there is some plausible hypothetical pathways that it might work. It's probably not a very large effect if it does work, because those previous studies should have shown that.
Thank you.
Question from the YouTube chat channel from Jerry Krasner. He asks, is reinfection a concern? And if so, what are the implications for antibody duration and vaccine lifetime?
Yeah, that's a good question. So is it is reinfection a concern? Absolutely. Because we get reinfected with coronaviruses throughout our whole lifetime of the regular circulating ones. And the reason that happens is because our antibodies to those viruses go away relatively quickly. What we can say with this particular coronavirus is that if somebody gets the coronavirus and they get really sick, they mount really good antibodies. So the antibodies that were made, the antibodies that were used to make those monoclonal antibodies were from people who got really sick and almost died. Some people in other did die. But people who have asymptomatic infection or just get mildly sick, their antibodies do not last. Now, the real question is, how much antibody do you really need to protect somebody from reinfection? And that is still not known. And in these vaccine studies, as they go forward, that's something they're looking at very closely. It's the correlative protection is how much neutralizing antibody response do you have to protect. I will take a small divergence here, is that antibodies aren't always good. For some viral infections, antibodies can actually be bad, known as antibody-dependent enhancement, where the antibody latches onto the virus and brings it closer into the cell to that ACE2 receptor and allows it to cause infection. We know that that happens in some viruses like RSV and dengue being the most famous in this area. And coronaviruses have some of that ability. I don't think ADE is a big player in the pathogenesis of CoV-2, but we need to keep in mind that being a risk as we do these vaccines that stimulate the antibody response.
Well, we have a question from Mike Houghton again. If Mike can turn on his mic and ask the question, because I'm not sure what he means. So I might learn something here.
Yes, thank you. My question is very simple. Can you address herd immunology and how it works?
I misread it. It's hard.
Herd immunity?
Yes.
Yeah, so that's a good question. So herd immunity actually is what I was trying to go for in terms of how many people need to be protected in a population to make the epidemic die out. So for SARS-CoV-2, which spread for every one person who's infected, if the whole population is susceptible, you would expect 4 people to get infected. And then those 4 people would then spread it to 4 other people, and it just gets out of control, which is where we are now. But if you use various protective measures like masks and quarantining stuff, you can decrease the number of those contacts to decrease that spread. Vaccines could also do that, or immunity in general, or therapies, and you could decrease the number of susceptible people in the population so that not everybody is susceptible. If you could get to 3/4 of people in the population to be protected, then you should be able to have that epidemic go away. So that's our goal. The problem with that is if the FDA says that we need a 50% vaccine, you would need a 50% vaccine, everybody to take it, you still don't get to 3/4 of the population protected. So you'd still have to do things like masks and quarantines and need some treatment. So, and even then, you're never going to get all of the United States vaccinated, right? There's the last survey that I saw is that only 1/3 of people would be willing to get vaccinated. So herd immunity is based on vaccines is probably not attainable.
Thank you.
I have a question from Ed Fernandez. Edmund? I'll ask the question for him then. Can you speak as to how we're handling mutations of COVID-19? I think you partly
How
addressed
we're,
before.
how we're handling mutations?
Yes, how are we accounting for them, I suppose, in the development of therapies?
At the moment, we're not. People talk about it a lot, but they haven't really accounted for it on the vaccine side or on the treatment side. I do know that in my trial, we are measuring them, and we can see that some monoclonal antibodies select for certain mutations. We don't know if that has a clinical significance yet. So in other words, if somebody was to have a virus that became resistant or develop a mutation that reduces susceptibility of that drug, do they do worse? It's likely that they do, but we don't know yet, and that's something that is ongoing research-wise. On the vaccine side, nobody's accounting for it because we don't know what the correlate of protection really is for these vaccines.
Yeah, and we don't know if the epitopes that they're using are ones that are susceptible to mutation or not. So
yeah. I am sure that those epitopes will become resistant, but whether or not it impacts the vaccine effectiveness to a clinically significant degree, I don't know.
We have a question from the chat box. Joshua McLean would like to know, is there any indication that the virus shows signs of being man-made? You can skirt
man-made, yeah, the man-made question. I think it's always a healthy discussion, but no, I don't think that there's any evidence that the virus was man-made. And the reason I say that is we knew bats, so this virus came from bats. We knew bats had they still do, have tons of coronaviruses. And there were researchers who went and sequenced all these coronaviruses from bats. And there are some that are 98, 99% similar to this virus. So this virus was hanging out in a bat for a long time, and then it had the opportunity to jump over to humans probably a few times. And through that iterative process, it adapted to us, mostly likely through that ACE2 receptor. And that's how the game got started. And we see the same process with all these infections. HIV came from chimpanzees through repeated exposure. Zika and dengue are both closely related, and they came over as well. And measles, it's just down the line. Zoonotic, and this is what happens. Zoonotic infections.
So we have a question from Frederica de Rema. She can answer the question. Frederica.
Yes, the question is, what about the remaining long-term effects from people that quote recovered? And also, is there any thinking of treating these people in some way so that the effects get mitigated and not kind of flaring up? We don't know if that happens also eventually.
Yeah, Frederica, thank you for that question. That's a super good question, because I work in a COVID clinic here at UCSD where we see lots of people who have lingering symptoms, and some of these symptoms can be quite debilitating in terms of fatigue, mostly fatigue, but muscle aches and mental cloudiness, etc. The first part of that, for treatment-wise, I think the treatment needs to happen early. So we need to knock the virus down as soon as possible, and that's the plug for my trial, is that's exactly what we're trying to do. And by doing that, we think that we should be able to stop a lot of those lingering symptoms. We actually don't know why some people have these long recovery times and lingering symptoms, and I have some patients who've been pretty sick for a few months now. They never got so sick that they needed to be in the hospital, but now their symptoms are lasting months after the infection is good and gone. It's just the virus caused this damage, and the body hasn't yet recovered. I do not have any treatment to give those people. People have looked at steroids and anti-inflammatories, etc. And there's anecdotal evidence that some of those work, but there's no clear therapeutic benefit yet.
So Gail Milstein has a question. Gail?
Yes, if you've had COVID, would you still need to get vaccinated?
Good question. I don't know. What I can say is that if you had COVID and you were really sick, you're more likely to be protected than you were if you didn't get very sick at all from the first infection. But honestly, I don't have any idea of whether or not that initial infection is going to protect you long term, or whether or not the vaccine might even work better for you. It might be like a booster to really get your immune system in working order to protect you. Along those lines, I don't actually know what's going to protect us. Is it B cells? Is it T cells? Is it our innate immune responses? It's clear that if the better you are at making interferon early, the more protected you are, versus if you make interferon late, which is actually a bad thing for the hyping up the inflammatory response.
I'm going to close with a couple of questions of my own. One is, given the difficulties of doing these clinical trials with large enough numbers and controlled enough way for them to give meaningful data, to what extent are these trials purposefully targeting a particular point in the etiology of the virus? So you pointed out that, one drug acts only at the beginning, it seems, and it's not very useful to give it late. Are these trials stratifying the patients and looking at what time is the right time to give the drug, so we know, so we don't miss efficacy, by drowning it out in administration at times when it's not very useful?
Yeah, that's the difference with what happened early versus what's happening now. So the trials that are being conducted now very much encapsulate exactly what patient population that is useful for a particular drug. And that's the reason there are those 5 active trials. There are inpatients and outpatients, people who are sick, people who are not that sick, people who are before they get sick, people who have clotting disorders, to really hone down on, this is the theoretical mechanism that we're trying to intervene on, and this is the patient population that should benefit from that.
And I guess I wanted to ask a general question about testing, which is no tests are perfect. And there are a large number of tests out there, whether they're serological or looking at genomics, meaning there are tests offered by different vendors. Do you have information on what the false positive and negative rate is in general for the gene tests for presence of RNA of the virus?
Yeah, we could spend way too long talking about tests, and I have lots of opinions about them. I guess in broad strokes, I don't think the serology tests are very useful at the moment at all. They don't tell you if you have an active infection, and they're not very useful for telling you if you're protected or not. So I very much do not like those tests. I think they give people either false security or false worry. So that's one side. The other ones are the active molecular tests. So if you have an active infection, it's the detection of the viral RNA, or it's the detection of the viral antigen. And for the most part, the viral RNA tests, the PCR-based tests, are much more sensitive and specific than the viral antigen tests. The viral antigen tests can be pretty sensitive, but they definitely can't be very specific. And in saying that, I just want to make sure that people know it's sort of a wild, wild west out there for diagnostics. There are so many companies that have their own tests, and each of those have different specificity and sensitivity for the population that they test. And the reason that's important is that I want a very sensitive test to rule out infection. I want a very specific test to rule in an infection. So if somebody comes to my emergency room and they have all the symptoms of COVID, I want to use a very specific test to say, oh, you have COVID. Or let's say I want to get my school open and I want to make sure that everybody who comes in doesn't have infection. So then I want a very sensitive test. And not all those, and every single test has a different sensitivity, specificity. And the only point here is that if you're a workplace, or you're a hospital, or you're whatever, and you're going to get your test, you need to know, is this test better for what I'm trying to answer my question for?
I think the other thing that I was really asking about is all tests have their problems. So a very sensitive test is likely to produce false positives.
That's right.
Even in PCR, which is notoriously able to amplify anything that will hybridize to a primer. So if you're testing large numbers of people, you're going to have this problem of a significant number of false positives or false negatives. And I have not been able to get any information on some of the leading tests on what that rate is. And I know I looked at early results for the Abbott test, and they had, I think, 4,000 tests they ran, 4,000 samples, and they had, it was like perfect results. There were no false positives, no false negatives on all the samples they ran.
Yeah, so I would just really caution people to understand that those companies play that they rig the books, and the way they do that is they know kind of what their false positive and false negative rate is on mixed samples, and then they go out and test in a certain population of people who have low rates of disease, right? So if you have low rates of disease, you want a very sensitive test to pick up those that are few out there. But that means you're going to increase your, you're going to worsen your specificity, so you get more false positives tests, right? So these companies cook the books a little bit to make their tests look like they're 100% sensitive or 100% specific based on what population that they bring in. If you had a group that had 50% of COVID at the time, and you used a test there, you want a very high specificity test and not have any false negatives, right? But the prevalence in the population changes both the sensitivity and specificity. And that's the problem for all these tests. My advice there is that somebody, the FDA or the US government, needs to have quality controls and not leave it up to the companies for their diagnostic tests. They need to put out blinded panels with certain prevalence and rates within that blinded panel, and then have the company give them the results. And then that would tell you what the sensitivity and specificity of that test is. But there is no standardization for that at the moment.
It sounds like a good job for NIST.
Yeah.
That's what they do.
Yeah.
Anyway, I think we'll close with that. Thank you so much, Davey, for giving this very informative lecture. And thank you everyone for joining us. The recording of tonight's lecture will be available to everyone on the PSW Science YouTube channel, where you will be able to see it at any time. So please share the link with your friends and subscribe to the channel for notifications on new postings. And with that, I think we can say goodnight to our speaker and bid him adieu.
Good night. Thank you, Larry. Nice to meet everybody.
Before you go, there are just a few important notes that I want to share with you. Our next lecture will be in 2 weeks on October 9th, 2020. The speaker will be Eric Verdon. His lecture will be on aging, focusing on molecular mechanisms of senescence and interventional approaches that may enhance life as we age and may in addition extend normal life expectancies. Eric is a professor of medicine at UC San Francisco, and he is the director of the Buck Institute, an institute dedicated to the study of aging. We hope you will be able to join us for this lecture. The rest of the fall lecture series has been posted to the website. Please check there often for updates. Last but not least, we want to thank tonight's crew, James, Robin, and Ann, for putting together this broadcast. Oops. And with that, I will now adjourn the 2425th meeting of the Society and wish everyone a good evening. The meeting is adjourned.